Okay, we ended up on the last recording talking about those three aspects of the dinitroaniline herbicides: the volatility, orange color, and But on this one, we want to start back. We got to finish up with dithiopyr. So that we'll just refresher what we just looked at. We're worried about the formation of the spindle fibers is what we're concerned about here, and that's what dinitroaniline herbicides and this one pyridine herbicide we're going to talk about, which is dithiopyr. Okay, and dithiopyr is in a different family, but it pretty much does the exact same thing, except remember it was a microtubule associated protein that actually inhibited, not the it was not the binding to the tubulin itself. Okay, so we have the family pyridine. Its common name here is dithiopyr, and it's sold as dimension in turfgrass. We're dealing with the same type of setup with as far as pre-merge, selective herbicides, residual. But this one does have some early post-emergent activity on crabgrass, typically around one-tiller crabgrasses. So that early post-emergence within the first 4 to 6 weeks of crabgrass germination, you will see activity from a post-emerge standpoint. But this is, it's an odd artifact of this herbicide. It's typically applied in that standard pre-emergence application timing that you have to apply this herbicide prior to germination occurring. So remember that. So we have to apply it before the weeds actually germinate. Okay, so that ends up on all our mitotic inhibitors. So the dinitroaniline inhibit tubulin, which stops the spindle fiber formation. Dithiopyr inhibits the microtubule associated protein, which stops spindle fiber formation, but they essentially are doing pretty much the same thing. Okay, let's go back to cellulose. Now we talked about cellulose being this biopolymer, and that is enzymatically connected by cellulose synthase. So we know that any herbicides that actually can block cellulose synthase can actually block the formation of cellulose, which is essential for cell wall formation. We do have some families that do this. This is the herbicide isoxaben, which is in the benamide family. And it's selective in that we talked about the dinitroanilines and dithiopyr in that they will only control small-seeded broadleaves and grasses. Well, this one really only controls broadleaf weeds and really has much less activity on grasses. Similar to the dinitroaniline, it is a residual herbicide, root-absorbed, pre-emergent, contact in nature. And it does have selectivity as well. Okay, its use is as a standard pre-emergent herbicide, and the weeds that it controls again is only going to be broadleaf weeds. This is isoxaben, and just to walk you through a little bit about isoxaben. Again, we're talking about mode of action, cellulose biosynthesis inhibitor. Site of action, cellulose synthase. The family is going to be benamide. And it's sold as the trade name Gallery and sold in Snapshot in horticulture, and this is primarily where it's used. It's used in ornamental potted ornamental production. It's used in ornamental plant areas where you're going to have a lot of broadleaf weeds that are going to pop up in those areas. So this is a very good herbicide for that use, where you're trying to grow broadleaf plants, and therefore you need broadleaf selectivity. Unique, and many times it's combined with something like pendimethalin or a similar type of dinitroaniline herbicide. It's a very unique herbicide that has very little activity on monocot species and no post-emergence activity. Therefore, large-seeded broadleaf weeds, not typically controlled by DNA herbicides, dinitroanilines, can be controlled by isoxaben. We have another one that's actually the alkylazine family, and this one is very new, and it's referred to as indaziflam, and a very new herbicide, only released in the past couple of years. It is a It's very similar in that it's selective, residual, pre-emergent contact herbicide for the most part when utilized, but you can get root and foliar absorption with this herbicide, similar to what we discussed with the dithiopyr. Okay, standard pre-emergent herbicide and unique. And sorry, this is a misprint here. Sorry for that, but I'm almost done recording. We're not going to stop now. It controls both broadleaf weeds and grasses. Okay, for the most part, we're dealing with very broad, at least as far as these pre-emergent herbicides are concerned. This is probably the most broad spectrum in that you'll get some grass and some grass control and some broadleaf control as well. Okay, so if you just read the notes and you didn't actually listen to the lectures, you may have missed that. So hopefully you, well, obviously you are listening right now. So tell your friends, make sure they listen to the lectures. It is the mode of action for indaziflam is cellulose biosynthesis inhibitor. It inhibits cellulose synthase, that enzyme, family alkylazine, common name indaziflam, and is sold under the trade name now of Spectacle. I think others will be coming in the future, but right now sold under the trade name of Spectacle. Is a primarily pre-emergence control of grass and dicot weeds. It can injure cool season grasses, which is a negative. Therefore, it's usually only used in warm season grass turf, and that's where Spectacle is labeled. It's a turf grass herbicide right now. Ornamental uses are coming in the future, but right now it is a pre-emergent herbicide for turf grass use. Okay. And miscellaneous comment here, new pre-emergent herbicide for use in turf grass and ornamentals. This herbicide will likely be developed for use in crop systems. Normally herbicides are launched in crops and then fitted for use in turf grass later on. Indaziflam is one of the only herbicides that have been developed in the opposite direction, first to turf and then to crops. Okay. All right, so that covers our of our 6 major groups. That covers cell division, disruptors, and really anything that what we dealt with was mitotic inhibitors, things that are going to mess up mitosis, and we also dealt with things that inhibit cellulose synthase, which is going to stop cell wall biosynthesis. Okay, so that covers all of that group.